Hi everyone, my name is Anna Coffaro, and I am here to walk you through my semester project, which I chose to center around the natural history of crabs found in the Hawaiian Islands. So to give you a little bit of background, took a special interest in crabs about 5 years ago, ish, when I opened a macro photography book by Susan Middleton called Spineless. And in it, she documented all kinds of invertebrates, crabs amongst them, and they just fascinated me. Um, since I can't be in the field right now, I thought this would be a wonderful opportunity to get to spend time with crabs intimately through drawing. I also have a background in the arts, so I took the time to get to know them a little bit better this way. I'd like to dedicate this presentation to all people who love the sea, and to those who have, maybe a special affinity for crabs like me. Just feel like it's not too often that we get to see some of the unusual creatures in this group. We tend to see a lot of the things that just get eaten or sold in markets, but my intention with making this presentation was to bring up some of the things that are just new to us and unusual, and maybe fill in some of the gaps in my own curiosity. Um, Also, as an artist, I think it's an interesting place to question how scientific illustration now fits into the scientific schema, if it does, how it does, etc. So to start off, I'd like to give you a little bit of the early natural history. Some of the first most important collections of crabs in the Hawaiian Islands came from a bunch of scientific expeditions in the 19th and 20th centuries. We have a whole bunch of American ones right here, some important European ones. This image to the right is an illustration plate by William Stimson, who is one of the names I continually encountered. Stimson, Dana, Mary Jane Rathbun, Charles Edmondson. These are some famous authors, researchers, and scientists who did a lot of work in this field. I would like to highlight Mary Jane Rathbun, who I first encountered when I was working at the Smithsonian National Museum of Natural History. She was lovingly dubbed the crab queen there, and she was the institution's first ever female full-time curator of crustacea, and considered to be one of modern day's first female carcinologists or crab studiers. Here is one of these specimen jars that she labeled. One of my jobs is to go through and label all of these and organize them and digitally input them, etc. So here's one of the trapeze crabs she took under her wing. Some of the modern contributors of this project include Dr. Peter Castro, who's just, wow, the trapezid king, has written numerous pieces about trapezid, trapezid crab phylogeny and systematics. And then Keoki Stender, who was actually a former mopper here at UH and is this wonderful scuba guru who has this wealth of marine life images that he shares on his website, and has collaborated with some of the scientists I'm going to mention, as well as the Waikiki Aquarium. But to turn back to Peter Castro, one of the works I examined of his was a catalog of Hawaiian crabs, and in this catalog he documented 68 species of Anomura and 284 species of Brachyura. Just to clarify, Brachyura are considered true crabs. They've got that small tail and thick exoskeleton, and Anomura tend to be the more unusual formed crabs, such as this albatross hermit crab. But for the sake of this presentation, I'm just going to focus on one group called the trapezids, which are also known as guard crabs. Here's a little bit about their taxonomy. We are focusing on family Trapezidae, specifically of the genera Trapezia. And as I mentioned, they're called guard crabs because they have a special symbiosis with their coral in which they guard it. They are symbionts of this host coral, and how this works is the coral provides shelter, safety, and food, and the crab in return protects it from invaders and predators such as the crown of thorns, helps with water circulation, Um, I'd like to point out the work of Dr. Chelsea Consul, who is one of the scientists that came to my coral and coral reef class. Considering these are coral guard crabs, much of her work focuses on them. And what she found is that they have extremely aggressive behavior. One of her studies documented this type of behavior towards other commensal organisms that might try to enter the coral. So they were really inhibitive of their arrival and persistence and responsible for very negative encounters with them, just showing to demonstrate, or just trying to demonstrate how dependent they are upon them. They were so hard to study that she actually had to knock them out with clove oil in order to remove them. They were that attached. But this type of aggression also exists in their mating habits. So trapezes are some of the most aggressive mating crabs that there are. They usually begins with a period of display followed by aggressive fighting, very high level of injuries. Um, this was just, again, to illustrate how dependent they are on their host coral. They really want to prevent anyone else from getting in there and anyone else from mating in there. So taking a look at their biogeography at large, we know that trapezes inhabit this tropical Indo-Pacific region. I've highlighted that Indo-Malayan / Coral Triangle area, which is, very traditionally regarded as one of the most prominent hotspots from which species derive and further speciate. Taking a look at the Pacific Plate, 32 species of trapezes are known to inhabit this area, but very low levels of endemism. So this is kind of a questionable thing because typically, like I mentioned, that Indo-Malayan region is pretty much held responsible for or is regarded as a center of diversification. But in one of Casper's papers that I read, he mentions two researchers who question this because there's such an unusual distribution pattern amongst this group. They actually think it might be the result of accumulation of species from separate sources rather than the singular center of dispersal. But I'd like to highlight the Eastern Pacific part of the plate, which has very low diversity, as well as the Hawaiian Archipelago, and then that Southeastern Pacific part of the plate, which has much higher diversity. So if we take a look at the Hawaiian Archipelago, only 6 species of Trapezia have known to have been recorded here. Only 1 out of that 6 is endemic to the area, Trapezia intermedia, which is the most common trapezid crab. But we ask ourselves, why aren't there more? And researchers, they do account for the fact that maybe there are more present, but they're just so rare and seldom collected that, we don't know, such as Trapezia rufopunctata, who's, very rare. But really, considering how isolated Hawaii is as a landmass, we know that that larval dispersal distance is super important. So in order for crabs to really make it that far, they have to have that larval longevity and long distance within them. Sometimes they could reach the island and just have unsuccessful colonization on the corals, but in general, we know that they face a lot of dispersal barriers, including temperature limits, land barriers, that water distance. So in general, most of the diversity of trapezes is correlated with that shorter length of dispersal distance and shorter larval development stages in general. Another huge barrier is that Eastern Pacific barrier, which I'm highlighting right here, which attributes to the fact that there's much greater diversity in areas like French Polynesia and that southern plate because they don't have to travel past that barrier. So in addition to that short larval dispersal distance, we also know that there's higher diversity of host corals in this area. There's both acroporid and polyporid. So yes, that reduction of isolation plus the high host diversity equals higher trapezid diversity. We in general know that trapezid larval stages and dispersal are not super well documented or studied. It is proposed, though, that coral rafting might be responsible for transporting those juvenile stages beyond those limits. So in general, Castro highlighted that much of this geographic distribution can be attributed to sympatric speciation. So that's characterized by large number of sympatric species that cohabit with endemics in peripheral regions. Excuse me, that was my timer. Just to go back a second, sympatric speciation is the evolution of new species from a surviving ancestral species, so that purple region right here. And while both continue to inhabit the same geographic region, overlapping in ranges. So these smaller circles right here indicate areas where you have that speciation occurring, and you might see some of the low-level rates of endemism. Now, I want to highlight that trapezids tend not to have host specific tendencies, as we do see several species have been found sympatrically living on different areas of a single colony. But what we do see is mate selection and recognition playing a significant role in that sympatric speciation. So there's evidence that species-specific behaviors and chemical detection that are involved in mating might help maintain that reproductive isolation and help explain some of that speciation. So this was a cool term for me. Assortative mating is a mating pattern in form of sexual selection where individuals with similar phenotypes mate with one another more frequently than would be expected from a random mating pattern. So very concretely, this example of Trapezia digitalis, they were attracted to different color variants within that species. But anyway, this was just evidence that, suggested that this could lead to that reproductive isolation amongst these different species. Most interestingly to me was the significance of color variation amongst trapezids. So due to that, or as a product of that sympatric speciation, they look a lot alike. The colorations are very similar, and that makes identifying them extremely hard. And I accurately identifying them. So in the records that I looked at, we have a lot of those early contributors to the research, Edmondson, Simpson, Dana, Rathbun, actually mistaking some of these guys for misidentifying them essentially. What I'm trying to highlight here is that color pattern and morphology is almost more significant than than some of those body plans themselves, because they tend to really overlap, and it's super important to recognize these differences when you have a live specimen, if you want to accurately identify them. This series of images right here is actually the same species. This is Trapezia digitalis, but as you can see, there's some variety, but there's also lots of overlap, between these guys right here, these guys, and it can be really, really confusing. So essentially what we need to do is increase our molecular studies in order to elucidate just how they speciated. And if they're continuing to form new species, there's the possibility that these slight variations in color patterns might actually reveal that more species are diversifying. So this was, I even noticed this when I was looking at the species myself. This is an example in Trapezia tigrina. I mentioned digitalis earlier, but here is the exact same species. We just see these color polymorphs within species that make it super confusing, because, I look at this and I think this could very well be another one that I'm thinking of, such as Rufo punctata. But I need to wrap up, and so I'm just going to formally introduce to you these 6 that we see here. This is Trapezia tigrina, otherwise known as a red-spotted guard crab, often confused with Rufo punctata, as I previously mentioned. And essentially, over the next couple of slides, I'm going to just document for you where they're found throughout the Hawaiian Islands. I detail their coloration, a bit about their sizes. Here's Trapezia bidentata, which is often mistaken for one of those earlier species Trapezia semidoce, which I mentioned. Rathbun actually had a hand in naming. So here we go, Trapezia pedentata, Trapezia intermedia. This is the most common guard crab throughout the archipelago. We have Trapezia flavopunctata, the yellow spotted guard crab. Again, Edmonton is very famously known for confusing this one with Trapezia rufopunctata, which is this guy, as you can see, extremely similar. This one's the rust spotted coral crab. There are extensive morphological notes on them in some of the literature that I read through, but here's another one, Trapezia digitalis, the brown guard crab that I mentioned to you earlier. So I made a little pictogram right here just to, look at where we might be able to find them. That was sort of one of my hopes in doing this, was to create a bit of a graphic, since we are here as students, where we might be able to find them ourselves. So I'm going to leave it at that. I'm going to post this as well as the second half of my crab catalog illustrations that I documented and explored. But here are a bunch of some of my resources. Again, I'd like to highlight Peter Castro, Chelsea Consul, Kyoki Stender as some of the key contributors to this project. Thank you all so much. If you have questions, I'd love to talk more with you about them. I absolutely cherish these crabs. They were the first loves of my life in terms of my affinity for decapoid crustaceans. Okay, everyone, stay safe. Thank you all again, and hope you enjoy.